Hello everybody, welcome to the last lecture of Unit 3 and our last lecture of the Experimental Techniques in Molecular Biology series. So we're going to come all the way to the contemporary time now. We'll end this lecture with a full explanation and illustration of systems biology. But really the theme of today's lecture is going to be about DNA binding proteins and how to identify and measure their binding of DNA. That's going to start with a more old-fashioned, maybe perhaps a bit antiquated experimental approach called gel shift assays. We'll get a little bit more high-tech and talk about chromatin immunoprecipitation, still a single gene experiment or a small number gene experiment. We'll go all the way into systems biology and talk about ChIP-seq, which combines chromatin IP with sequencing to get more of a systems biology approach. And again, we'll end this unit with a full illustration of systems biology and what we mean by that in the modern age. So again, to put this into some context and to frame this lesson or this session, we want to think about DNA binding proteins and what they do. This brings us back to Unit 1. We talked about HDACs, HATs, mediator, general transcription factors, regulatory proteins. These are all examples of DNA binding proteins, at least TF2D with its TATA binding protein subunit of the GTF binds to DNA. And wouldn't it be nice to be able to see what proteins are bound to, or in the case of mediator, recruited to specific DNA sequences? In other words, we can ask the experimental question, is TF2D present? Is the pre-initiation complex there at the gene promoter? Is the gene on? Or is something like TUP1 present? Is a repressor there? Is the gene being turned off by the cell? So being able to detect which proteins are bound or recruited to specific identifiable DNA sequences, and to also measure when they are bound there, allows us to answer a lot of different molecular biology questions that really get at the mechanics of gene function. Reporter genes, microarrays, they give us answers about when genes are on and off. They give us the is a gene on or off, but they don't give us the how or the why. They don't tell us which regulatory proteins or signals are present at that gene's promoter. What we talk about today gets to that level of detail. Which regulatory proteins are bound to which genes, when, and why. So the theme of this lesson is how do we measure proteins binding to specific DNA sequences, and how do we do that both in vitro and in vivo? So our first question along that line is, how does a specific protein specifically bind to DNA at all? We've talked about this both explicitly and implicitly all semester long. We talked about an example like ROX1 binding to the promoter region of hypoxic genes. Well, how does ROX1 bind to the promoter region of hypoxic genes and not other genes? More recently, we talked about restriction enzymes, recognizing specific 6 base pair sequences and cutting somewhere in those sequences. How in the world does a restriction enzyme bind to those 6 bases and no others? When we get at non-specific binding, something like DNA polymerase, it's easy. If we give a protein a positive charge, it'll stick to anything that's negatively charged, including DNA. But we're not talking about non-specific electrostatic binding here. We're talking about specific sequence recognition. How do cells, or proteins, achieve that? So in other words, how does a protein distinguish one DNA sequence from another and bind to one and not the other? The easy and short answer to that question is structure equals function. The shape of each particular DNA binding protein, along with their charge and other chemical properties, allow them to precisely fit into and interact with specific DNA sequences. The more detailed answer is most DNA binding proteins cannot separate double-stranded DNA into single strands, and so the way that they're accessing DNA bases is through the potential of non-covalent interactions. They're doing it through chemistry. Each particular sequence of DNA gives a uniqueness to the overall property of the DNA in that region, and that chemical property, or set of chemical properties, can be probed and accessed by DNA binding proteins through the major groove of DNA. The major groove of DNA is the larger, more accessible break or separation in the backbone of the spiraling staircase of the DNA helix. So when DNA helicizes, when it coils, when it twirls along its axis, it does so a bit unevenly, and we have these alternating major and minor grooves. The minor groove of DNA is quite shallow and narrow, and the major groove is deep and wide. So every other face of a helical turn is a major groove where the chemistry of the bases can be accessed. So this is looking at DNA kind of laterally. If we look at it more vertically, we see here's a major groove and a minor groove, a major groove and a minor groove. And on the other side, we have minor groove, major groove, minor groove, major groove. So this is one helical turn here, one 360 twirl of the DNA double helix. And we see that we have a minor major and a major minor on that one helical turn. So to say it simply, the major groove of DNA provides a window into the DNA sequences that can be accessed and measured. So it's almost like we're peering in from the outside at the bases, or maybe even more accurately, it's almost like we're reaching in directly, reaching in and touching the bases without actually disturbing the DNA and getting a sense, a chemical sense of what the DNA sequence is by that touching. So, in essence, this all relies around the potential for non-covalent binding forces. So what we're looking at here is a very small piece of a transcriptional regulatory protein, a DNA binding protein in green, and it's reaching into a specific DNA base. This is a TA base pair, and it's reaching in through the major groove. And one amino acid of this transcriptional regulator is making non-covalent contacts or non-covalent bonds with the adenine base. In fact, we're looking at two hydrogen bonds. We have a hydrogen donor from the protein interacting with a hydrogen bond acceptor on the adenine. And we have the opposite. We have a hydrogen bond acceptor on the protein interacting with a hydrogen bond donor on the adenine. So these are two non-covalent forces and two non-covalent bonds that are possible between the DNA binding protein and a base pair in the DNA sequence. So really what these transcription factors are doing, what all DNA binding proteins are doing when they bind to DNA with specificity, is they're taking examples of these interactions, but they're stretching them along many, many sequences. So if we imagine that the next base is a guanine, and another amino acid of this protein is interacting with the guanine through different non-covalent forces. And then we think of another base along that sequence as a thymine, and the transcription factor has an amino acid that can interact in that thymine. Then what we have is kind of this mirroring, this I hate to use the word complementarity because we always use that with DNA, but we have this complementing of non-covalent force potentials in the DNA sequence itself, made up by the sequence of bases, and a mirroring, complementing potential for the same non-covalent forces and non-covalent bonds on the binding surface of the protein. So DNA binding proteins use every non-covalent trick at their disposal to interact specifically with DNA bases. They use hydrogen bonds. They also use electrostatic forces, ionic bonds, positive and negative charges interacting. They use hydrophobic interactions, van der Waals interactions. Essentially, many of the things that you've already learned about in your chemistry classes are being used non-covalently for DNA binding proteins to recognize specific sequences. All of this is happening at the edge of the bases, there at the major groove, through chemical forces. Each of these individual interactions between a single amino acid on the transcription factor and a single base in the DNA, each of those individual forces, each H bond, each ionic bond, each van der Waals interaction is weak. But if we get a long enough sequence with many, many non-covalent bonding potentials, and we get a long enough surface of the regulatory protein that mirrors those bonding potentials to actually create these non-covalent bonds, then we've got lots and lots of individual weak forces that taken together creates a relatively strong interaction. And although you might not recognize it right away, we have a perfect analogy for this in the regular real world. Imagine each DNA binding protein can make 4, 5, 6 contacts with any random sequence of DNA. That's not enough to hold it there because it's weak, but if we sum those up across a specific DNA sequences, and now we have that happening at many, many bases, we're looking at 20 or 30 non-covalent contacts strong enough to keep it there. So what is the real-world example of this? The real-world example of this is Velcro. If you're ever geeking out to the extreme and you're really, really bored, you can do what I once did and look really closely at Velcro. One face of Velcro is made up of very small plastic hooks. You can actually see the hooks if you look closely enough. And the fuzzy, the soft part of any Velcro interaction is just that. It's fuzz, but those fuzz is actually looped threads, looped fibers. So each of those kind of fuzzies is a loop, a thread that goes out of and back into the Velcro face. And the way that Velcro works is that each one of those hooks, those plastic hooks on one face of the Velcro, hooks into a loop on the fuzzy side of the Velcro. Now, if you want to get really, really lame, you can actually take a single hook, isolate it, kind of bend it out from the others, and you can get that single hook to loop onto a single loop of Velcro, and you can pull those apart, and they come right apart. There's no real force there at all. It's like the slightest bit of resistance, and it gives immediately. That one interaction is very weak. But now take one face of that Velcro and smush it up to the other, and try to pull those apart, and now it doesn't feel weak at all. It's like, got to really pull to separate those two pieces of Velcro. It's not that the force of any single interaction was any stronger when you smush the two Velcro pieces together. Quite the opposite. The force of each of those individual Velcro interactions was exactly the same as that little force of the single Velcro interaction. But so many of them create such a large summative force that you really have to pull to tear those two Velcro pieces apart. And that's exactly what's happening with specificity and DNA binding. What we're seeing is that these regions of the DNA binding protein, here in gray, each region is making multiple individual weak contacts non-covalently with the major groove bases of the DNA. Like those individual Velcro interactions. Very, very weak and transient. But if we get enough of a face of a surface interaction between amino acids on the DNA binding protein and complementing non-covalent forces coming from the bases of the DNA in the groove, now we have a true Velcro interaction, an interaction of large force. So as we kind of look across this face, we see that this alpha helix number 4 is making about 3 contacts with this AT and this GC here. A little bit of Velcro. The next alpha helix 3 is making what looks to be about 4 or 5 interactions with this T, and this G. So that's more Velcro. This alpha helix number 2 is making 1 or 2 interactions. Alpha helix number 1 is making 1 or 2 interactions. But what I want you to appreciate is that whatever non-covalent interaction potential is here, whether it's a negative charge, a hydrogen bond donor, hydrophobic force, a van der Waals, is being perfectly complemented by this specific sequence in the DNA. Maybe it's the other charge, a hydrogen bond acceptor, a complementary hydrophobic face, or a van der Waals interactor. And so this surface of this transcription protein, transcriptional regulatory protein, will bind only with a high number of interactions, with a high number of forces, with this sequence TCT, AG, AT. Change any one of these bases and you're going to lose some of this Velcro, you're going to lose some of this bonding potential, and then that will not be enough force to keep this protein bound to that. So again, structure equals function. The structure of this protein creates a face of bonding potential that is perfectly mirrored by the specific DNA sequence it has evolved to bind to. Change that binding face or change the DNA sequence, and you'll lose the required number of non-covalent interactions to make this binding possible. This protein binds to this sequence with specificity. Amazing. So this provides the strength and the specificity of binding because the number of interactions needed to actually hold the DNA there, the number of Velcro interactions needed to hold the DNA there, is only given by this specific sequence. So every DNA binding protein is unique. It has its own specific interaction face that recognizes its own specific DNA sequence. But that said, life has evolved a relatively small number of structures to hold DNA binding surfaces. You can think of these as scaffolds for binding to DNA. We call these domains, DNA binding domains, and they come in a limited number of flavors. So almost like cars, right? We've got almost an infinite number of cars, shapes, sizes, makes, and models, but they're all basically structured the same way. 4 wheels, 2 axes, a chassis, the engine, right? Very, very similar form, but lots of nuanced differences. We see the same in DNA binding proteins. Each specific DNA binding protein structure is either called a domain or a motif. The difference between domains and motifs will probably be made clear to you by a biochemistry professor, but if you'd like, you can find me on YouTube. I have a domain motif tutorial there on YouTube, or I'm happy to go over it with you if it's something that's confused you before. Some examples of these domains or motifs are homeodomains. Homeodomains is a specific configuration of protein subunits or protein secondary structures which come together to form an interaction surface. And then the actual identity or chemistry of that interaction surface, hydrogen bond acceptor, hydrogen bond donor, charges, hydrophobicity, van der Waals, all of that is going to change dependent on the specific sequence that homeodomain has evolved to bind to. Zinc finger is another example of a commonly evolved DNA binding motif. Leucine zipper is another. There's others that are not listed here. For example, ROX1 belongs to a family called HMG proteins, which have their own unique DNA binding domain. So again, vastly different strategies that proteins use to bind to DNA, and then specificity comes from the actual amino acids that are present in these domains, giving those interactions in the major groove. Again, these are the most common eukaryotic DNA binding domains and motifs. There are thousands among transcriptional regulators alone. Now, some of these proteins also dimerize, meaning they pair up. So two individual subunits, two individual polypeptide chains will come together and interact non-covalently with each other to create a larger protein made up of two subunits. These can be homodimers, where the two subunits are identical, or heterodimers, where the two subunits of the dimer are different. And since each subunit of the dimer recognizes its own unique, smaller DNA sequence, this approach of making dimers allows DNA binding proteins to modularize their DNA binding. What do I mean by that? So here's an example of a DNA sequence that we're considering. We'll make it double-stranded so that we can see that it goes in both directions. And let's imagine one subunit binds to GTCT. So the interaction face, the DNA binding domain or motif of this protein, can reach into the major groove and make specific non-covalent complementing interactions with the bases GTCT in the major groove. So we bind there. There's enough Velcro between this protein and this sequence to hold this protein to this sequence non-covalently. So we'll have that binding. If this was a homodimer, we could have this dimer now recognize specifically the sequence GTCT-GTCT. Because the subunits are dimerized, they travel together, they will only recognize GTCT twice in a row in tandem. So it would only bind here. This would be a homodimer. But a heterodimer could form that recognizes a different sequence, for example, CGAC, GTCT. By making this heterodimer and having these two subunits travel together, now the specificity of the 8 base pair site that they bind to has changed. So we can mix and match these individual subunits and create dimers that recognize larger sequences that are different than the individual sequences each subunit could recognize alone. So by mix and matching these subunits, we can get our complexes, our dimers, to bind to specific longer DNA sequences and achieve their binding with more specificity. So that was quite a digression, right? So in order to understand when and how we measure DNA binding to DNA, we really did need to understand how DNA binding proteins bind to DNA with specificity. And we wanted to drive home this point of non-covalent binding, the major groove, and the idea of this Velcro, individual weak forces summed together across a DNA binding interface allows a protein to be recruited to and stick to specific DNA sequences. But we still haven't answered what I said this lesson was about. I told you that we were going to talk about ways in which, in the lab, we can measure protein binding specifically to DNA sequences both in vitro and in vivo. And while we might now understand conceptually how DNA binding proteins bind to DNA sequences specifically, we don't understand anything about how we might measure that. So let's get into the experimental analysis of DNA binding. We'll talk first about gel shift assays. This is a completely in vitro approach, all artificial in a tube. We'll talk about chromatin immunoprecipitation, which is a more in vivo approach, and then again we'll get to this systems biology thinking when we talk about ChIP-seq. So first, a gel shift. The take-home message that we want to always keep in the back of our mind is that the purpose of a gel shift assay is to measure a DNA binding protein binding to a specific DNA sequence with specificity. So to do that, we obviously need the protein that we are hypothesizing is doing this binding. And we need a DNA sequence, so an artificial double-stranded DNA molecule that contains the sequence we are supposing our protein binds to. And we call that DNA molecule a probe because we are probing the ability of the protein to bind with that DNA molecule. And we label that probe in such a way that we can make it visible. Sometimes this is done with radioactivity, and that radioactivity will expose radioactivity-sensitive film. We'll generate a true exposed film of this. We could do it with fluorescence in ways that we've talked about before. We could do it kind of immunologically with antibodies or chemically with some kind of conjugated enzyme. Any way that we can make this probe visible, we can use for this application. And what we can do is we can add that probe to the well of a gel. We can perform gel electrophoresis here, and we can run that gel, and we would see that that DNA probe runs very, very low in the gel because it's a fairly low molecular weight. It's a short piece of DNA. It's not very long. It's going to move quickly through the gel. And so we can see that is running there because we've made the probe visible. And again, it runs low in the gel because it's relatively small. And then we could repeat this experiment where we add our probe to the reaction as well as our protein. And remember, our hypothesis is that this protein binds to this DNA with specificity. And if that protein does in fact bind to that DNA, some of that probe will be shifted up in the gel. Hence the name of the assay, gel shift assay. We will shift some of that probe up higher in the gel because the protein and the DNA together is larger and a higher molecular weight than the probe DNA alone. And we get that gel-shifted pattern. So when we develop the film, we would see something like this. Some of the probe is not bound by the protein, so we still have a free probe band here at the bottom, but because our protein binds to our probe, we've shifted some probe up and we see that band, a higher molecular weight band as well. And maybe at this point, we high-five, we jump around, we do a big yahoo, because look, we proved our hypothesis correct. Our protein binds to our DNA of interest. Well, don't get so ahead of yourself. What we've shown is that this protein will bind to the probe, but we really haven't answered anything about specificity, have we? So we want to do some controls. Our first control is going to be to repeat this experiment yet again, but this time we're going to add our protein, our probe, and our probe DNA some more, but we're going to add this as a cold competitor. In other words, we're going to add the same exact sequence of DNA that we see in our probe, but this new addition is not going to be visible. It's not going to be radioactively labeled. It's not going to be fluorescent. Whatever we did to the probe to make it visible, we're not doing to this competitor. And if the DNA is binding specifically to our DNA and not some other contaminant that's in the tube, this cold competitor should remove the shift. We should see this pattern. What's happening really in the tube, this on the right is what we would see on the film. We're peeling back the curtain here on the left, is that yes, our protein is binding to DNA, but because we've added specific cold competitor, and we've added it in such a higher concentration of our probe, most of the DNA in the tube is actually cold competitor. Remember, by cold we mean invisible. So even though our protein is binding to DNA and it's shifting that DNA, that shift is not visible in our gel because all of that DNA is cold, invisible. And since the probe itself is so outcompeted by this cold, invisible DNA, we see a restoration of that free probe band there. So adding more evidence that, yes, our protein was binding to that sequence. But a critic can come up to you and say, at this point, all you've really proven is that your protein binds to DNA. Like, what if your protein is just overall positive charge and it's sticking to DNA non-specifically? It'll stick to any DNA. Because essentially what you've shown me here is that if you add labeled probe DNA, your protein sticks to it and shifts it up. And if you add unlabeled excess unprobed or unlabeled DNA, you can lose that shift, but your protein's just sticking to DNA. So you're going to do another control. In this control, you're going to add your probe, which is visible, your protein, which binds to the probe, but now you're going to add non-specific competitor. You're going to add excess DNA that is unlabeled, but this DNA is going to have a completely different sequence to it. So make the prediction. if your protein is binding to the probe DNA with specificity, what's going to happen here? If your protein is binding to the probe DNA with specificity, in other words, if it's recognizing a specific sequence that's in the probe, and that sequence is not in this cold non-competitor, then your protein is going to ignore that cold competitor completely because it doesn't have a recognition sequence on it. In other words, the binding potentials in the major groove of this cold competitor aren't right and don't match our protein, so our protein will never stick to it. So our protein will ignore that DNA and only bind to the labeled probe, and that should give us a shift again. However, if your protein was binding to DNA non-specifically, and the critic was right, your DNA is just positively charged, your protein is just positively charged and it's binding to any negative DNA it finds, then this mutant non-competitor should also outcompete the shift. Just like we saw in lane 3, your protein should prefer that other DNA just as well, bind to this other DNA, and lose the shift. So if we continue to see a shift in lane 4, that suggests even more that your protein is binding to that particular probe sequence and not any DNA randomly. Sounds good. Now the critic comes back and says, you know what? Still not convinced. What you've shown me is that a protein in the tube is binding to the probe with specificity. You haven't shown me that your protein is binding to the tube in specificity. In other words, I don't trust your biochemical skills, and I think that when you did this protein prep, you didn't just get your protein, you got some contaminating protein in the background too, and it's that protein that's binding to your probe with specificity, not the protein you're actually studying. So you do one more control experiment. And here you add the probe, you add your protein of interest, and you're going to add an antibody against your protein of interest. And if it is indeed your protein of interest that's binding to the probe, the addition of the antibody should create what we call a super shift. This is the highest molecular weight complex loaded on the gel. It consists of the probe, clearly, because we see a visible band, your protein binding to the probe, and then the antibody binding to your protein. You've almost made this 3-decker sandwich of probe, protein, antibody. And that being an even higher molecular weight structure will give a band even higher in the gel. And now you have shown that your protein binds to this probe sequence with specificity. A really nice example, not only of measuring DNA binding and of gel shift assays, but also the contortions that experimental biologists and chemists must often put themselves through to disprove any alternative interpretation of their data and only have their conclusion fortified by the evidence of the data. So here's an example of that very type of experiment. Again, just to show you that, this isn't just a conjecture, this isn't make-believe, that this is done in labs. So we have a very similar thing. The probe here is binding at the bottom, very, very dark. Most of the sample is probe. When we add our protein of interest, we get the shift. When we add the antibody to our protein of interest, we get the super shift. Some additional controls were done here that we don't need to go into, but the same general idea. So that's a gel shift assay. Very powerful, right? We can conclusively prove that a protein of interest binds to a specific DNA sequence with specificity, but it's all in vitro. It's all artificial. It's outside of the physiological context of the cell. Just because a protein can bind a sequence in a test tube doesn't necessarily mean that protein binds to that sequence in a living cell. So in order to get a more in vivo approach and begin to be able to measure and understand how and if and when proteins bind to specific DNA sequences in a living cell, we turn to chromatin immunoprecipitation, or ChIP. So again, ChIP is going to allow us to detect a protein being bound or recruited to DNA within a living cell at a time and under whatever condition we choose to subject those cells to as the experimenter. So how is ChIP different from a gel shift assay? Well, basically it comes down to in vitro versus in vivo. Gel shift assays are very artificial. It has no opportunity for cofactors or other complex members to be involved in the binding or recruitment. ChIP is all in vivo. Whatever proteins can do in a living cell, they are doing in that living cell, and we can measure it by virtue of a ChIP. And also, if we think of things like mediator, mediator doesn't bind DNA specifically. Mediator binds to RNA polymerase II. RNA polymerase II is bound to TF2D. TF2D is bound to the DNA. So this is a recruitment to DNA by association, right? Mediator is actually 3 steps removed from actually binding to DNA directly. But very much so, mediator is recruited to DNA, and that's physiologically important. No subunit of mediator would ever bind to any DNA we throw at it in a gel shift assay because there's no direct interaction between mediator and DNA. But in ChIP, we can see that mediator is recruited to DNA because ChIP allows us to see all proteins. Well, ChIP allows us to detect a protein of our choosing being recruited to DNA under whatever condition we're studying. And before we go any further, this is chromatin immunoprecipitation. So if this is an immunoprecipitation experiment, that means we're going to be involving antibodies. Those antibodies are going to be pulling things down in the tube, enriching for targets. So keep that in the back of your mind as well. So here's how ChIP works. What we have here is nucleosomal DNA. So each of these gold balls or beads is a single histone core with DNA wrapped around it, right? When did we talk about that? How many weeks ago? And then we have 2 examples here. We have this single green protein. It's a single subunit, single polypeptide, binding directly to DNA. And then we have this dimer. Looks like a heterodimer to me, dark blue and purple. We have this dimer binding to DNA. And it's recruiting this red protein. So we essentially have 3 DNA binding proteins and 4 proteins being recruited to the DNA here. We've got the green protein binding as one, and we've got the heterodimer binding to DNA recruiting the red protein. Very first step of chromatin immunoprecipitation is cross-linking the sample with formaldehyde. Formaldehyde is going to induce covalent bonds between all proteins and DNA, well, period. So anything that's near anything else is going to chemically cross-link to that next thing. Histone proteins are going to cross-link to each other. Histone proteins are going to cross-link to the DNA in the nucleosome. The purple protein is going to cross-link to the blue protein. Purple and blue are going to cross-link to the DNA. Red's going to cross-link to purple and blue. Green's going to cross-link to everything is covalently glued to everything else. Formaldehyde glues everything together through chemical cross-links. And then we're going to bust it all up. Either with sound energy, such as sonication, or an enzymatic digestion, we're going to shear this DNA up into relatively small pieces, somewhere on the order of 200 to 500 base pairs in length. So everything gets broken up into pretty small individual fragments of chromatin. And now we do the immunoprecipitation. We're going to add an antibody that specifically binds to and recognizes whatever protein we're studying. And in the case of the green protein, we would add an antibody to the green protein. In the case of the red protein, we can add an antibody to the red protein. We can add a single antibody to any protein we're interested in studying. And the question we're really asking here is that protein recruited to DNA when we cross-link the cells with formaldehyde? When we hit the cells with formaldehyde, was the green protein bound to a specific region of DNA? Was the red protein recruited to a specific sequence of DNA? So this antibody is really at the heart of the ChIP assay because it differentiates and distinguishes what protein we're studying. So we add that antibody, it binds to our protein of interest, and then we add what is typically referred to as resin. Resin is like a very, very fine granular sand, usually made up of complex carbohydrates, and we can cross-link different proteins to that sand. So in the case of a chromatin immunoprecipitation, the protein we would cross-link to this granular sugar sand would be a protein called protein A. And protein A binds to the opposite non-business end of an antibody. And because it's this granular sand, these resin molecules are relatively heavy in the grand scheme of everything else we're working with, and they will come down to the bottom of a test tube quite readily. In fact, if we leave the test tube on the bench for half an hour, this will come down in just 30 minutes, which shows just how heavy it is. But if we spin it down in a microfuge, it'll come down in a few seconds. So as we look at the bottom of the tube after we do one of these spins, what do we see? We see resin. Okay. So that's these granular carbohydrates. But what's covalently linked to the resin? Protein A. Ah, okay, great. What's the protein A binding to? It's binding to the back end of the antibody. Interesting. What's the antibody bound to? The antibody is bound to whatever protein we were interested in studying. And what's that protein covalently linked to because of formaldehyde treatment? It's covalently linked to the DNA that it was bound to when we hit those cells with formaldehyde. So what we have actually immunoprecipitated are these small chromatin pieces, these small DNA fragments that were bound to our protein of interest when we hit those cells with formaldehyde, and that's what we're interested in. So at that point, we wash that immunoprecipitate a couple times, just make sure we get rid of anything that was there non-specifically. We reverse the cross-links, so now we liberate the DNA from everything else it's stuck to. We isolate that DNA, and then we're going to probe for its presence using PCR. So we're going to add primers to our specific DNA of interest, and we're going to ask the question, is this PCR-able? Right? So if we're doing an AMB1 immunoprecipitation, for example, if we're doing a ChIP experiment on the gene AMB1, and maybe we chipped for ROX1, if we think back on that example of coordinated regulation from a couple weeks ago, ROX1 as a transcription factor binds to the promoter of AMB1. So now imagine we cross-link those cells. ROX1 was bound. We glue ROX1 to the AMB1 promoter. We shear everything up with sonication. We add an antibody to ROX1, and we immunoprecipitate that antibody with protein A resin. Once we liberate the DNA from that immunoprecipitation, we've got AMB1 promoter in that tube. So now we add primers to the AMB1 promoter, and we ask the question, can these primers amplify anything? In other words, is the AMB1 promoter in this tube as template? If the answer is yes, if we can generate a PCR product with AMB1 primers, that means the AMB1 promoter was present in that tube. And the only explanation for that was that AMB1 promoter sequences were immunoprecipitated by an antibody for ROX1, and that means ROX1 was bound to that AMB1 promoter when we hit those cells with formaldehyde. So here are some examples of that. So this is a MOT3 chip. MOT3 plays a role at the AMB1 promoter as well. You can see that we are testing for the presence of the AMB1 promoter. We heard about MOT3 in one of the papers that you did read earlier on in the class, but in this case we're chipping for its presence. And we see that when the AMB1 gene is being repressed, MOT3 is bound to the AMB1 promoter. This is PCRing for the presence of the AMB1 promoter, immunoprecipitated by MOT3. So the fact that we have a dark band there means that we were able to PCR the AMB1 promoter when we immunoprecipitated for MOT3 protein. MOT3 protein was bound to that promoter when we did the chip. After 1 hour, 2 hour, and 4 hours of hypoxic growth, when the AMB1 gene is on, we see that MOT3 signal is way, way down. MOT3 is no longer at the promoter, and we no longer see its presence. And we have controls here, right? We've got input controls that basically show that we had DNA present, chromatin was present for each of these immunoprecipitations. It's not like there was any degradation here or anything. And we have an untagged control that shows that if we don't have an antibody immunoprecipitating anything, we don't get any DNA in the bottom of the tube. So this is an old-fashioned chip. This is a chip from a paper that I was lead author on. But ChIP is done a little bit more high-tech now. This is from the paper that you are reading this week. Here we're immunoprecipitating for centromeric proteins and actually looking at the impact of histone variants on the centromere formation. So we've got this kind of sliding scale of PCR amplicons, of PCR primers. Region number 3 is a PCR product right over the centromere. And then region 1 and 2 are upstream of the centromere. Region 4 and 5 are downstream of the centromere. So we kind of create this sliding window of PCR products, and we can look to see where proteins are bound. And what we see here is that all 5 of these proteins are bound to the centromere specifically, and their signal for binding drops off precipitously as we move away from the centromere in either direction. So we're able to detect protein binding to regions of interest using chromatin immunoprecipitation. Now the question is this systems biology? If systems biology is looking across the entire genome, if systems biology includes looking at what happens at all genes, at all DNA sequences, and taking this snapshot of a living cell all at once, did we do that here? And the answer is no. As powerful as chromatin immunoprecipitation is, it still only allows us to look at one gene at a time. Now, it's true, we can interrogate immunoprecipitated chromatin for whatever sequences we want, but we have to use specific PCR primers to do that. In other words, if I wanted to look at other regions that MOT3 was bound to, such as the heme13 gene, I could do that in this experiment, but I have to use PCR primers specific to heme13 in order to detect that MOT3 binding. If MOT3 is binding to some other gene sequence that I'm completely unaware of, ChIP will never allow me to spot that because it's not systems biology. I can only detect MOT3 binding at regions I specifically look for. I'm not looking across the whole genome. So just like reporter genes only allow us to look at one specific gene at a time, ChIP only allows us to look at one specific DNA region at a time. And just like microarrays allow us to look at the expression of all genes simultaneously, ChIP-seq allows us to look at protein binding to all DNA sequences across the genome. It's a whole genome analysis. So we are officially entering the world of systems biology here, and what a wonderful way to wrap up Unit 2. Gel shifts, super powerful. I mean, conclusive, unambiguous detection of protein binding to DNA. And ChIP, being able to do that in vivo is just incredible. But how many genes do these techniques allow you to study? Only as many genes as you are probing for, either with a visual probe in gel shifts or PCR primers in ChIP. We now refer to these kinds of studies as well as reporter genes as single gene studies, and some molecular geneticists will even snicker at this, like, she's doing a single gene study, because we don't have to do these kinds of studies anymore. We can now do systems biology. Cutting-edge studies look at the entire cell, the entire organism, the entire living system in one experiment. But if we're going to do this, if we're going to look at a single living system as a single whole, if we're going to take snapshots of everything that's happening in a living system at any one time, we need specific techniques and also specific computational power to do that. And that's why systems biology really is its own subdiscipline. So we've covered a technique that does that already. Again, microarrays takes an entire comprehensive snapshot of all genes being expressed at all times in that cell system. Microarrays study all of the genes being transcribed by the cell or collection of cells whenever we did that experiment. That is unequivocally systems biology. But by the end of this lesson, we'll also see that ChIP can be modified in such a way to have a systems biology approach as well. And when we end this lesson, we'll also give greater definition of systems biology as a discipline. We'll talk about the genome, which is really the first systems biology concept even before systems biology existed. And then we'll talk about more recent innovations or considerations, more recent concepts, such as the transcriptome, the proteome, and even the interactome. All right, so let's return to ChIP now, but modify ChIP so that it fits a systems biology approach for studying DNA binding across the genome. So as you can probably already figure out, ChIP-seq is really chromatin immunoprecipitation followed by DNA sequencing. And by coupling those two technologies, we can look at the DNA regions across the genome that are bound by a specific DNA binding protein at any time, genome-wide. Starts off the same way as a traditional ChIP. We have our DNA, and we have our protein of interest, and our protein of interest is bound to or recruited to our DNA. We hit that with formaldehyde, we cross-link everything together, and then we shear that chromatin up with either enzymes or sonication. So now we have this fragmented genome, this fragmented set of DNA molecules with our protein of interest bound to some of them. Again, this is done to cells in their entirety, so everywhere our protein of interest was bound to across the entire genome is represented in this sonicated, sheared mess, just like a regular ChIP. And now we immunoprecipitate. We add an antibody against our protein of interest, still no change, and we're going to precipitate all of the DNA that's bound to our protein of interest to the bottom of the tube using protein A resin. And then we're going to reverse those crosslinks and isolate the DNA. Again, identical to ChIP. We haven't done anything different yet. Here, if this was a standard ChIP assay, we would have in the bottom of the tube DNA representative of all the sequences across the whole genome that our protein of interest were bound to, but the only thing we would be able to detect are the things that we add PCR primers for. So in other words, like I said before, if there was a specific genomic region that we had no idea our protein of interest was bound to, and we would have no reason to add primers to that specific sequence, we would never detect it. What we do in ChIP-seq is instead of going straight to PCR, we take all of these DNA molecules and we use them to prepare what's called a sequencing library. I've dropped that term library before, not too long ago. A library is a heterogeneous collection of cloned DNA fragments. So essentially what we do here is we take each of these individual DNA molecules that was immunoprecipitated here, and we clone them individually into vectors, into plasmids. Same general way that we talked about before. Some technical differences, but none worth noting. So now we have a collection of plasmids, each of which contains DNA that was immunoprecipitated by our protein of interest. That's our sequencing library. And then, using big dye, second gen or third gen sequencing, usually either big dye or second gen, we can sequence each of these inserts in this sequencing library. So what we're doing now is we are determining the sequence from scratch of every single DNA molecule that our protein interest was bound to when we cross-linked those cells. And sometimes we're going to get lots and lots and lots of the same sequence. And that means that our protein was bound to that sequence in lots and lots and lots of places. And other times we're going to get unique sequences that our protein is bound to not so often, representing places that our protein is not bound often at. And this allows us to generate a DNA binding genomic map, such as what we see here. This is a single chromosome. Well, this is one chromosome, this is a second chromosome, and here's a third chromosome. So we're looking at different chromosomes that our protein of interest is bound to. And the size of these peaks represents how many of this same identical sequence did we pick up in our sequencing reaction. And that is a measure or indicative of lots and lots of protein binding with high efficiency. And all of these shallow peaks here represent regions that our protein was bound to, but not at such a high level. So we see that our protein is bound in some intermediate way here at the end of the chromosome. It's really down to the level of probably statistical noise here throughout the chromosome, but at the beginning and beginning two regions of the next chromosome, we see lots of our protein binding, and then not too much, and then tons of protein binding on these chromosomes here. So we get, again, a genome-wide survey of where our protein of interest is bound to across the entire genome at the time at which we cross-link the cells. To try to say it more simply, we are detecting every single place in the genome that our protein was bound to when we hit those cells with formaldehyde. That includes regions we suspected our protein was bound to, and regions we had no idea our protein was bound to at all. This is systems biology. It's surveying the entire genome. It's giving us a snapshot of protein of interest binding everywhere in any one time. Amazing, amazing, amazing technology. So let's wrap this unit up by just having a full appreciation of systems biology and the kinds of things that system biologists consider and do. So again, systems biology really started with the notion of a genome. Genome is the sum total collection of all the genes scattered across all the chromosomes of our genetic composition. So here we see a human karyotype, 46 total chromosomes, 23 pairs. The 23rd pair is asymmetrical or non-homologous. That's because this is an XY or male individual. This is a genome. All of the genes here in one image. And studies that look at the entire genome are systems biology studies, whether they're microarrays, whether they're ChIP-seqs. When we are asking questions of what is happening here, what is happening here among all these 46 chromosomes, that's systems biology. But we now consider another layer of systems biology that we refer to as the proteome. And if the genome is the sum total of all genes, the proteome is the sum total of all proteins. Now, proteins fall into classes such as metabolic proteins that are responsible for making and managing energy, signal transduction proteins, which are sending signals, allowing signals to be sent and received around the body. We've got membrane proteins, of course, the transcription factors that we've talked about before. Some proteins are involved in apoptosis or programmed cell death. We've got proteins that add phosphate groups for regulation called kinases, proteins that take those phosphate groups off called phosphatases. We talked about DNA methylation proteins when we were talking about the restriction enzyme and modification system. So all of those proteins considered as a single whole is the proteome. And we know a lot about the human proteome. We've done a great deal of work in the last decade and a half studying the entirety of human proteins as they exist in the context of a living cell or a living organism. So again, systems biology, not studying individual proteins, studying all proteins. There's also a subset now that studies the transcriptome. And this is the idea of all transcribed regions across the entire genome. Now, not surprisingly, most transcribed regions are protein coding genes, protein coding RNAs or messenger RNAs. So of the messenger RNAs, most of those messenger RNAs are true protein coding messenger RNAs. But we also have some weirdo examples such as long non-coding RNAs, they're transcribed. Pseudogenes, things that used to be genes but evolved to not be genes anymore, are sometimes transcribed. We've got short RNAs of unknown function that are definitely doing something important, but we're still not sure what they are yet. So even in the transcriptome, there's a lot of ambiguity, a lot of black boxes that we haven't fully deduced. And systems biologists who are studying the transcriptome are studying the entirety of the transcriptome, all of the transcribed regions of our genome. There's also the metabolome. So rather than studying individual facets or pathways of metabolism, systems biologists who are studying the metabolome, who are engaged in metabolomics, are studying human metabolism as a whole. This image that you're looking at, this kind of circuit board looking network diagram, is actually the sum total of eukaryotic metabolism represented in one schematic. Anything and everything that happens with energy management, that happens with ATP production, biosynthesis of molecules that we need to survive. If it is metabolic, it's represented on this schematic, which is incredible that we even understand all of metabolism. And for those of you that have already learned about biochemistry at some detail at the undergraduate level, there's an aspect, a central pillar here that you might even find recognizable. It's there in the middle of the diagram near the bottom. I'll blow it up here. This is the citric acid cycle, the hub of all eukaryotic metabolism. What's coming into this citric acid cycle is glycolysis and the pyruvate dehydrogenase complex, for those of you that have learned about that. Again, the central hub or the central core of all metabolism, but every single aspect of metabolism is represented in the metabolome, and it is now being studied as a single systems biology process. Love it. We talk of the cancerome. The cancerome includes all of the genes and proteins that are responsible for contributing to cancer. That includes the proteins and genes that protect us from cancer, tumor suppressor genes, as well as the proteins and genes that can cause cancer to progress. Now you might ask yourself, why in the world would we have in our genome genes that cause the progression of cancer? It's unavoidable. At different points of our life and different points of our development, we've needed aspects of cancer in order to survive. The problem is we don't need all those aspects together. I'll give you an example or a set of examples. We have genes for proteins in our genome that just cause massive cell proliferation. These proteins just scream signals to cells. Grow and divide, grow and divide, grow and divide, grow and divide. Sounds like a tumor, right? That's what cancerous tumors do. They grow and divide, grow and divide. But of course, every single one of us started off as a single-celled zygote, and 36 weeks later, we came into the world as somewhere between a 6 and 8 pound baby. That required a mind-bogglingly large number of cell growth and proliferation. So we needed those genes and those proteins during embryogenesis to drive that rapid cell growth to get to a healthy baby, but we never need those genes again. They lie in our genome. They've been silenced for those of us that are healthy, but they're still lurking in our genome, those proteins. And if they become active, they start screaming that signal again. Divide, divide, multiply, multiply, grow, grow. Those are oncogenes. We also have genes for proteins that cause cells to migrate, that cause cells to grow, move, migrate, and disperse across our body. We need these genes to be expressed in our immunological cells. White blood cells need to leave the bloodstream and enter wound sites to protect us from infection. Those cells should have those genes on, other cells should not. Now imagine a single gene that picked up mutations that not only turned on the embryonic proliferation genes, but also turned on the migration genes. Now you have a cell that grows and divides with high proliferativeness, but it also can leave the bloodstream and migrate to new places. Now we're talking about a metastatic tumor. So again, these individual properties are things that we need in different cell types at different times. Migration, proliferation, etc. What's the problem with cancer is that we have individual cells that have mutated in such a way to pick up all of these properties in one cell, and that's what becomes dangerous. The cancerome studies this as a cohesive whole in a systems biology approach. In fact, the cancerome field has identified virtually all of the genes that play a role in cancer and have categorized those genes through function based on what they do to cancer cells and why. We even have the interactome. The interactome is the full understanding of every single protein that has the ability to interact with any other protein. What you're looking at here is actually a three-dimensional network diagram. When you're looking at this live on its website, you can zoom into this. Each node or circle on this diagram is an individual protein, and each line connects that protein with another that it is able to interact with. So we now understand we're aware of all protein-protein interactions across the entire cell. Again, this is the interactome. And the fact that we are aware of this and can interrogate this and start asking the questions, why do these proteins interact? What's the physiological basis or role of these interactions is just astounding. So yeah, I mean, to me at least, this is mind-blowing. I cannot fathom the fact that we can do this, and we do it every day now. It still seems like science fiction to me, but it's not. This is science reality. For those of you that are interested in a world in research, who are thinking about getting a master's degree or a doctorate, these are some of the things that are on the edge of the envelope now of what we can do in the lab and the kinds of questions we can ask and answer, all through the lens of systems biology. So what did we talk about here? Well, we started off talking about DNA binding proteins in general. They bind to DNA specifically by making these non-covalent interactions with bases in the major groove. Whatever non-covalent bonding potentials are possible in a specific DNA sequence, those are mirrored and are complemented by opposing forces in the interaction surface of the protein. And the protein can now interact with that DNA specifically and have a sum total of weak interactions that provides enough force to recruit that protein to that region, very much like the Velcro analogy. So these non-covalent interactions are made only with that specific sequence of bases in that specific order, allowing enough energy of interaction to be generated to keep that protein bound to that sequence. And then we moved on to some experimental approaches or considerations. We would like to be able to detect DNA binding proteins actually binding to the specific DNA sequences that they bind to. We can do this with gel shifts or with ChIP. Gel shifts are in vitro, ChIP is in vivo. We reviewed how gel shifts can conclusively and unambiguously determine that a protein is binding to a specific DNA sequence with that specificity, but it takes a number of controls, such as a cold specific competitor, a cold non-specific competitor, and even an antibody to give us that super shift. We moved on to ChIP as the in vivo approach to DNA binding to specific sequences. Here we cross-link everything in the cell together at any one time. We shear that cross-linked chromatin up into smaller pieces. We immunoprecipitate against our protein of interest, but that brings down to the bottom of the tube the DNA that protein was bound to when we cross-link the cells. We reverse the crosslinks, liberate and isolate the DNA, and then we can detect that DNA with PCR. Powerful and interesting, but not systems biology. We defined systems biology, and then we used ChIP-seq as our example of a systems biology approach. Here we start off the same way, but once we capture the immunoprecipitated DNA near the end of ChIP, we don't PCR for it, we clone it and we sequence it. And that shows us all of the locations that our protein was bound to when we cross-linked the cell, giving us a snapshot of the entire genomic region that our protein is bound to across all chromosomes. True systems biology. And then we closed out with some of the most prevalent example of the omes, the genome, the proteome, the transcriptome, the metabolome, the interactome, as examples of where systems biology is now at in our current place in experimental biology. So I think I made it clear. Stuff blows my mind. Hopefully it blew your mind as well. This marks the end of Unit 2 and the end of our experimental approaches to molecular biology. After the exam, we'll move on to human diseases and begin to see the light at the end of the tunnel of the course. So until then, you know what to do. Let me know what we should be reviewing on Friday, and I look forward to seeing all of you then.